Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, as you know that in our previous session we talked about types of a plant cell. Under the series we would be conducting numerous lectures. So far we have conducted one lecture and that lecture was on plant cells and tissue structure. Today also we are going to talk on this very topic, that is a plant cell and tissue structure, and for this we have again with us in our studios Dr. Ashish Kumar Nandi. Dr. Nandi is professor in School of Life Sciences, Jawaharlal Nehru University. So dear friends, let's take advantages from his experiences and let's try to understand more about the plant cells and tissue structures. So without wasting any time, I would like to welcome our guest, Dr. Ashish Kumar Nandi. Dr. Nandi, welcome to the EDUSET lecture.
Yeah. Okay, thank you very much. I will continue from the place where I left in the previous class. In fact, I am discussing about the different type of cells present in plant and eventually their composition which makes different type of tissues which are present in the plants overall. See, plant is a living organism which needs to do many functions, and for that it is composed of many different types of structures. There are structure which gives it support, there are structures which enable plants to do photosynthesis, and there are structures which enable plants to take nutrients from soil. So, all these different kind of structures have a diverse kind of cellular organization, and it is really important for the students to know and very interesting also to, as a perspective of a biologist, to see how plants organize to generate different kind of structures. In the previous class, I have discussed how a plant cell looks and how a plant cell is different from an animal cell. I have given the emphasis on specifically like cell wall structure, which is so diverse compositions, but is also uniquely present in plant cells, but not present in animals. Besides that, I have spoken about centriolar organization, which is absent in animal cells, which is present in animal cells, but how cell division takes place in plants very precisely without having the centriolar structures. And we also discussed about the presence of plastids, their origin, and the division of cells. So, today we will discuss the type of cells you have in plants. So, if you look into the plant cell types, there are three basic types of plant cells which many of you or most of you are aware about of. One is called Parenchyma, another one is called Collenchyma, and the third one type which is called Sclerenchyma. These three structures of plant cells are very well described in the school level books. So, we will go through again to understand more, and we also learn lot of new things. Like Parenchyma, the Parenchyma, Collenchyma and Sclerenchyma, all of them have one word common with chyma. It could be Parenchyma, Collenchyma and Sclerenchyma. These words, all of them have the Greek origin. The chyma is originated from the Greek word enchyma. The enchyma means infusion. Something like a boiled tea can be called as an infusion of tea. So, you have a mixture of things, and infusion is the form by which cellular like cytoplasm or cytosol is looked as. So, Parenchyma is the one which is para means beside, it is something just poured in beside and nothing special about it. Collen, the Greek origin word colla. Colla means glue. So, the collenchymatous cells are having infusion with the glue in the sides. And scleren, the modern Latin word that originated from the Greek words scleros means hard and enchyma means infusion. So, if you think infusion is the cytosol or cytoplasm and having a scleros or hard covering is Sclerenchyma. So, as you can see, this Parenchyma, Collenchyma or Sclerenchyma, they are mostly distinguished by the way they are covered outside or cell wall. Like the structure you can see here, some cells like these ones have very thick wall, they are Sclerenchyma, and these small cells which are between the big ones have very thin wall, they are Parenchyma. So, today we will learn about these three type of cells, Parenchyma, Collenchyma and Sclerenchyma, and their main contributions in plants growth. These are the three different type of cells. If you make a cross section, you will find these are the Parenchyma having cytoplasm and nucleus. This is Collenchyma having thick cell walled cytoplasm and nucleus, and this is Sclerenchyma. Many students believe that Sclerenchyma are dead. This is actually not true. Like any other cell which die at the end of its life, Sclerenchyma do, but Sclerenchyma, many Sclerenchymatous cells life is shorter than the Parenchymatous cells. So, it is wrongly said that Sclerenchyma cells are dead. Nevertheless, they are very much alive, and one thing also I should tell you that any cell which takes bath, it takes bath like a Parenchyma, which is having a thin cell wall, and this Parenchyma cell eventually modifies into Collenchyma or Sclerenchyma. So, Parenchyma are the ones which are capable of dividing, they can divide, but once they get cell wall thickening, they become also resistant to division. So, they do not divide anymore like Collenchyma or Sclerenchyma. So, if you look into the differences between Parenchyma, Collenchyma and Sclerenchyma, there are many differences. The one I just told you is about division, like they are Parenchyma divides, but not others. But there are many other types of differences are there. Like this one I told you earlier, as just now I told you, cell wall thickening. In fact, the Parenchyma, Collenchyma or Sclerenchyma, the names are given based on the cell wall. So, no thickening, Parenchyma, thickening in the primary cell wall. This is something very interesting difference between Collenchyma and Sclerenchyma. There in the previous class I told you what is a primary cell wall and what is a secondary cell wall. Maybe I can show you a slide that I showed you in the previous class. Look at this structure. Here this is a structure of a secondary cell wall from Canada U fiber. If you look here, what you will find that this is the one which was made when cell was born. Immediately after cell division, the cell with which the wall with which a cell takes birth is called primary cell wall. Then the thickening of the cell wall can be outside the primary cell wall or can be inside the primary cell wall. Now, the secondary cell wall is the term used when deposition takes place inside the primary cell wall, and also quite very importantly, the deposition must be after complete expansion of the cell. The reason is that if a cell is not fully expanded and if it starts accumulating lignin kind of stuffs, they cannot grow more or they tear up in the pressure of expansion. So, secondary cell wall is the one that forms after complete cessation of primary growth. So, or cell expansion. So, this is where there is a difference between Collenchyma and Sclerenchyma. Collenchyma also have a cell wall thickening, but this cell wall thickening mostly as a primary cell wall thickening. In the sense, as the cells elongate, the cell wall thickening goes on, and quite interestingly you will see that this cell wall thickening does not kill the cells, because it is not throughout the cell. So, they do not tear it up, and this Collenchyma are very important for giving support to the young tissues like soft stems, leaf petioles and so on. On the other hand, the Sclerenchyma cells have a secondary cell thickening on the secondary cell wall. That means, the thickening which takes place is inside the primary cell wall, and that takes place when cell expansion is completed. There are more differences. Look at the shape. Most parenchymatous cells are usually round or close to round cells. In the other hand, if you look Collenchyma, most of the Collenchyma cells are elongated. These elongated cells are also sometimes Sclerenchymas are elongated. So, sometimes there may be confusion between Collenchyma and Sclerenchyma if you just look at the length, and many people prefer calling them combinedly as Prosenchyma or the type of cells which are elongated. But however, Collenchyma are usually quite elongated, and Sclerenchyma have a very diverse range, like they can be elongated like in vascular bundle, but there are many which are not elongated. I will tell you today. Mechanical strength, all of them, Parenchyma, Collenchyma, Sclerenchyma, all of them are capable of providing mechanical support, but the mechanism of support is different. In case of Parenchyma, the mechanical support comes due to turgor pressure of the cells. The cells are small, compact, and they are water filled. So, they give a lot of turgor pressure, and that turgor gives the support to parenchymatous cell. In contrast, Collenchyma cells are support come from the primary cell wall and Sclerenchyma on the secondary cell wall. Now, let us look where they are present. This is Parenchyma. This is again interesting point. Parenchyma is present in all the plants. Whatever the type of plants you take, it is not only the plants, the type of cells which are present in animal cells are also, most of them are called parenchymatous cells. Parenchyma actually is a very soft cell wall containing plant cells are parenchyma. Now, this Collenchyma you do not see in all the plants. This Collenchyma is present mostly in eudicots or higher dicots. This Collenchyma is absent in grass family plants or even in the ferns. So, many people or evolutionary biologists believe that formation of Collenchyma is an evolutionary event where, because of the plants have to get support from the land or they have to grow in land, they need a support, and Collenchyma evolved to give that support. Like say, if you talk about algae, I told you earlier in a class, algae are not considered as a land plant, and many biologists do not prefer calling algae as plant at all. They are completely aquatic, their body is soft, they do not erect against the gravity. So, algae do not need a supportive cell like Collenchyma or Sclerenchyma. Then the very early plant like Bryophytes. The bryophytes are land plants, usually is quite smaller, soft bodied, and they lack the vascular tissues. If you look interestingly, most of the bryophytes, or in fact all of the bryophytes, they do not contain any typical Celerenchyma cells. Some Celerenchyma like cells are reported in some bryophytes like Physcometrilla, but still they are not yet confirmed by the biologists that they are authentic Celerenchyma or not. So, eventually Celerenchyma is something, or Sclerenchyma. Sclerenchyma is also almost absent in case of bryophytes. From pteridophyte onwards, like the fern onwards, a few ferns have developed Sclerenchyma, and many ferns also lack Celerenchyma. Like again grasses, see many of the grass family plants are very strong, very big, and not very big, like some of them get very big, others like bamboos and sugarcane. Some grass family members are sufficiently big and grow, but they do not contain Celerenchyma. So, Celerenchyma is an evolutionary adaptation by the eudicots, and this Celerenchyma, actually this is a grass family. What happens, the Sclerenchyma develops very early. So, this Sclerenchyma which is present in the scattered vascular bundle, like the monocots have a scattered vascular bundle which can be scattered across the diameter of the cross section. So, those scattered vascular bundle supported with the fiber like Xylem fiber, Phloem fiber, they get the features like Sclerenchyma. Those Sclerenchyma cells provide support to the grass family plants, whereas most of the dicots, young plants, they rely on Celerenchyma. Like example, even if you think about a very big plant which is a perennial plant, the very shoot tip, but the very tip of the shoot or very tip of the root, they do not form the Xylem fibers, or their Sclerenchyma is not fully developed, but there a support is required, and that support is given by the Celerenchyma. So, if you look, or many of you who are aware of the secondary growth, you know that secondary growth is composed of the reformation of xylem and phloems, and the periderm which is made of cork cambium and cork cells. So, those, if you talk about cork cambium, living phloem, Phloem fiber, xylem fiber, that is the arrangement of the large trees, there no Celerenchyma. So, Celerenchyma is a typical cell which is not very abundant, present in certain type of cells, young tissues, to give support while growing. The difference is that Sclerenchyma cannot grow any further. Sclerenchyma get completely lignified, but the Celerenchyma can grow with having the thickening on it, because thickening is not complete, it is a partial thickening. So, that is how it grows. And if you look into their location, you will find that this location like in case of Parenchyma is mostly present in all plants. This arrangement of the table is getting bit disturbed, but location of this Parenchyma cells is present like ground tissues and bulk of the ground tissues and meristem tissues. And Celerenchyma, as I told you, is present in the as a hypodermal area of the young stems or soft stems, and Sclerenchyma mostly present in the fibers and vascular tissues. So, we will go one after another and learn about the details about them. Like Parenchyma, this is the they provide storage, metabolism and strength. So, Parenchyma does very important job for the plant. Like if you think storage, metabolism and strength, they are the three most important function of a plant, and that is carried out by the Parenchyma cell. So, you can see here, this is the cross section of a leaf. In this leaf cross section, you will find most of the cells are Parenchyma cells, and you know they carry very important job like photosynthesis, respiration and so on. So, this is are the mesophyll cells. So, you have something called spongy layer here and then you have palisade layer. So, these are the all parenchymatous cells and these are the ground tissues of the parenchymatous cells. Even the epidermal cells, if you look, they are also parenchymatous cells. And I told you a few minutes back, the very important feature of parenchymatous cell is that their strength come from the turgor pressure. This is a cross section of a leaf. Many of the leaves look very good strength is there, like they can take up the wind pressure. Many times you can find it it's very difficult to tear off. The very difficult to tear off is a different scenario, but even if you can tear them easily, they are quite strong, they can erect against the gravity. However, you will find many of the leaves, when they are slightly under water stress, the leaves collapse. Here is a photograph of a plant which is dried up. Here is a photograph of another plant that dried up. If you look, and you might have noticed, many plants, if they are dehydrated for a day also, they collapse. The reason for this collapsing is because their strength, the strength by which they hold their leaves against the gravity, that strength is completely lost when water is lost. So, these the parenchymatous cells main support, the strength support comes due to turgor pressure. If water is taken out, they droop. Parenchyma for metabolism, you see this is again mesophyll, leaf mesophyll, they are all green in color, they are filled with chlorophyll and then do photosynthesis. There is a term used which is called Sclerenchyma. This Sclerenchyma indicates the parenchyma that are filled with chlorophyll. So, they are not a separate type of cell, they are just a naming of another type of parenchyma that does photosynthesis. Water hyacinth, you know all the water hyacinths, they float, you can see them anywhere you go in countrysides. Now, how they float in the air? If you look here, what you will find, they have a very thick leaf petioles. These leaf petioles are filled with air sacs and that air sacs provide them the buoyance which help them to float in the water. If you make a cross section of a petiole of a water hyacinth, this may look like this. So, these are the large air sac like structures which is known as parenchyma. Please note here that parenchyma is not a cell, but a tissue structure of the parenchyma cells. This is sometimes the student do mistake by telling that parenchyma is a type of a cell. That is not true. It is a type of a tissue which is made up of parenchymatous cells, but they are composed of many dead parenchymatous cells. There is a difference again between the collenchyma, sclerenchyma and parenchyma after their death, because collenchyma is slightly or partially lignified, they shrink when they die or water is withdrawn. In contrast, sclerenchyma cells remain fixed and they do not shrink because they have a full lignification, and parenchyma can be shrinked in such a way that you cannot see almost at all, not there appears to be nothing left. So, many of you might have seen bamboo stem. If you cut a bamboo, this looks like this. You have a large hollow inside and there is a surrounding, there is a ring of tissue. That hollow structure of the bamboo is because of the death of parenchymatous cells, and those parenchymatous cells, once they die, you cannot see them and only you can see there are some scale like structure inside the bamboo stem. Similarly, here you can see when the water hyacinth petiole started or grown at the beginning, they were full of tissues like cortical tissues, and they are all parenchyma tissues. Later on they are developed to die. The word which is used commonly is called programmed cell death. They are programmed to die a death, they die, and then you cannot see them because their cell wall is very thin, which get compressed in the sides. So, then these structures are called parenchyma. This parenchyma formation is a stress inducible event. Like many times, if the plants are subjected to water stress or underwater, like then they develop parenchyma. This is an example of maize or corn. This is corn cortex. Normally corns are grown in the upland and without waterlogging. However, if you slowly waterlog for a few days with intermittent gaps in between, then you will find that maize roots develop this parenchyma. This parenchyma helps the waterlogged root to breathe and survive. So, this is an adaptation for the plants to survive under waterlogged conditions. Parenchyma for support. This is a tuber of a plant, potato. So, this is a potato tuber, and if you make a cross section, you will find these are the parenchymatous cells which just gather together to form a tissue which is quite strong. Many of you know that potato actually a quite hard stem, and there is full of only parenchymatous cells, and in between you can see this tiny purple dots, they are stained for starch. These are called amyloplasts. These amyloplasts are starch filled organelles which are present in large quantities in potato tuber. Now again I would like to bring your notice to certain things. Now look here, say these are the cells, this is one cell, this is another one, and this is another one. You can see a tiny air gap between the cells. So, if you have a roundish cells and if you put it anywhere which is stacked, there must be some gaps in between them. These are intercellular gap. They are often filled with some kind of matrix which are called as apoplast. This apoplast filled matrix is there in many parenchymatous cells. This is one thing. Now, you also let me tell you that many times parenchymatous cells need to be highly compact, very compact arrangement should be there. For example, many times it is believed that like meristem, in meristem the cells, parenchymatous cells are present in a very compact manner without leaving any intercellular space or any significant intercellular space. If you think of having a 3-dimensional structure to come together and not to leave any intercellular space, then that 3-dimensional structure is very difficult to imagine like a cube or cuboid. But having a structure closer to circular structure, globular structure, and leaving no air space is very difficult, like as it here. These structures are close to the globular structure, and now if you have to imagine these globular structures are closely arranged, but does not leave any space in between, then one has to arrange somewhere like this. This is a mathematical model, and this mathematical model actually gives a structure called tetrachydecahedron. This tetrachydecahedron is a structure where you have total 14 sides, and out of 14 faces, 8 faces are hexagonal and 6 faces are rectangular. So, there are 6 rectangular and 8 hexagonal faces are the closest to globular that can be compacted without leaving a gap in between. That is a mathematical model, very difficult to find in nature or very difficult to visualize in nature, and quite often if you isolate the cells and they modify the shape when you immediately as soon as you isolate them. Now, here is some cells which look to very close to have multiple solid surfaces, plane surfaces. This one is the closest you can think about of a tetrachydecahedron structure from a pith of a plant called elder plant, Samba stems. So, there Parenchyma cells are showing similar to what computer model had shown. Now, coming to Collenchyma. Collenchyma is another supporting cells. You can see the cross section of Collenchyma in the left side. You will notice here that unlike Parenchyma, these cells are thicker. Now, also look at something, that this thickness is not uniform throughout, in contrast to what I showed you earlier in Canada U fiber, which was a secondary cell wall. If you look here, this part is thick, this is thin, this is thick. So, for any cell it is not a uniform one, and this is a typical nature of a Collenchyma cells. These Collenchyma are, as I told you sometimes back, are largely present in soft stems. So, soft dicot stems or even petioles, they are supported with large number of Collenchyma when they grow. The reason also I told you, because Sclerenchyma formation takes place later and they cannot form as the elongating stem, because if they are elongating, Sclerenchyma cells tear it off, which happens in case of protoxylem. Like I told you also maybe in the previous class only, that if you take out a pumpkin or locky stem, petiole or stem, you will find there are large number of holes in between. Those holes are formed due to tearing of the protoxylems. So, protoxylems, they are lignified, and that lignified cells could not divide or elongate because of the lignification, and then when stem elongates under the pressure they tear it off. In contrast, Collenchyma can elongate having still the supportive thickening of the cell wall. That is the difference in the with the Collenchyma and Collenchyma. If you look at a structure of the celery cell, celery stem or any other stem, you will find the bunch of Collenchyma which is present just beneath the epidermis layer, and this is what quite often grow at the side of the phloem. So, this is a cross section of a celery stalk. These are the phloem fiber, phloem vascular bundles, and against each vascular bundle you will find there is a area which is filled with Collenchyma. This same structure which is shown here in exactly near the epidermis is shown over here, blown up. These are the epidermal layers and these are the phloems. So, these are the parenchyma cells, and phloem must be somewhere in the down to it. This may be the area where phloem started. So, parenchyma, this is parenchyma epidermal cells, in between the cells which are having quite thick cell wall and clump of thick cell wall which gives the strength of the stem, are Collenchyma cells. This is from a different plant. You can see here, this is Collenchyma, this is phloem. So, everywhere they grow where phloems are there. So, they usually are in support of the phloem, giving the strength. Now, if you look at the structure here, these are the ones which are all Collenchyma cells. The Collenchyma cells are longer, a clear distinction from the parenchyma. These are parenchyma cells which are roundish cells, and these are Collenchyma cells which are elongated cells. Look here, these elongations or stem thickening is not uniform. This is an enlarged figure of the Collenchyma cells. This is cell, Collenchyma cell, this is another Collenchyma cells, walls are thick in the side but look at the arrowhead. This is the junction between two parenchymatous cells, and this junction is quite thin, and this is what the characteristics of Sclerenchyma. They are non-uniformly thick, and these Sclerenchyma are highly metabolically active. They do many job in addition to strength. Many cases the Sclerenchyma are capable of doing photosynthesis. All the green petioles, green stems of celery which I showed you, they do actively photosynthesis, and that photosynthesis is mostly carried on Sclerenchyma. So, many times students wrongly say that Sclerenchyma are the ones which contains chlorophyll. That is not correct. I told you earlier, cola came from the words glue. Because they are glued in the side, the glue appearance due to the secondary primary cell wall thickening, they are called Sclerenchyma. This Sclerenchyma may not be uniform, sometimes may be uniform against a cross section of a plant. So, this is a cross section of a plant called Corposoma repens, in common name is called mirror bush. In the mirror bush, you have a uniform Sclerenchyma deposition, whereas in other plants like celery, and here it is called lovage plant or Levisticum officinalis, these plants you have Sclerenchyma thickenings in some part of the stems. So, whatever it may be, throughout the stem or some part, it provides mechanical strength. Now, coming to the last type of cells, what I told you as Sclerenchyma. When you eat naspati or pears, you feel some sand like things in your tongue. Like even if you eat them with your mouth, you will find some sand like particles, and which are quite hard. Now, what are these? Even this kind of cells are there also in supporters, and they are actually Sclerenchyma. So, the questions which I also discussed earlier, are the dead or the living, that we will discuss now. But let us see, I told you also earlier, like any other cell, Sclerenchyma are living, only thing is that they die little earlier than other parenchymas or Sclerenchymas do. So, wrongly people say Sclerenchyma dead, but that is not correct. I will show you today. These Sclerenchyma are broadly two types. Ones are called Sclereids, another are called fibers. These Sclerenchyma cells, quite often they are long like Sclerenchyma, and they club together, grow like a fiber. So, the fiber is not a type of cell. Quite often people say fiber is an arrangement of cells. Like most of the cases, fibers are made by mostly lignified, sometimes non-lignified thick walls, but usually they has to be long, and many cells club together to form a fiber. Whereas Sclereids are Sclerenchyma, but they are grown independently, not by clubbing. So, basically on the basis of clubbing, they are either called Sclereids or they are called as fibers. Look at this beautiful picture. This is a Sclereid in pear fruit. This is the stone cell, this is also called stone cell in the living, it is a Nespathy stone cells, and this is a micrograph of a stone cells. You look here, look at this arrow through which I am going. These ones are, or this one is primary cell wall, and this after the primary cell wall it got n numbers of secondary cell wall. So, there is a huge depositions of secondary cell wall, all happened inside the primary cell wall. Now, in spite of so much of thickening, this cell is still living. The reason is that you have cytoplasm, you have nucleus, and this cell, you can see this is the mitochondria. There are some mitochondria which are embedded in the, this is the mitochondria, no, these are ear membranes, sorry, these are ears, and these are some mitochondria, this is nucleus, this is cytoplasm. So, these cells has all of its living component. However, I must also say that the rate of metabolism is very low. So, it is not a highly metabolically active cell, this is a low metabolic activity cell, but living one. If you look carefully, a structure which is present in the side of the cell between two living cells, you will find a structure like this. This is plasmodesmata. That means, this cell not only doing its own metabolism, are also capable of communicating its neighbor with the help of well defined plasmodesmata structure. That means, they are very much living cells. This is a type of Sclereids, because these are not fibers, and this is called Brachysclereids or stone cells. This Sclerenchyma formation is also stress inducible event. Like many times you can see, this is a plant of a Bethula plant or bark plant. In the Bethula plant, this is the picture in the right side shows is area of Xylem parenchymatous tissue. These are all Xylem parenchymatous cells. You can see here, these are the natural Xylem fibers, Xylem parenchyma, and if you put a needle across the Xylem parenchyma, the cells which are surrounding those needles will convert into Sclerenchyma. So, Sclerenchyma formation is developmental, but quite often develop as a protective layer at the site of wounding, insect bite, pathogen infection. Many times while eating fruits like papaya, you would notice there are some hard cells between the soft papaya tissues. Those are quite likely made as a sclerotization or Sclerenchyma formation surrounding some kind of insect wounding or any kind of natural wounding. So, this Sclerenchyma formation takes place also against wounding or as a stress response. Look at this leaf in the left side, phylodendron leaf. This kind of leaves are very leathery and sometimes very difficult to tear off, but you can roll them easily. Some leaves do not tear off in the high wind, quite difficult to turn off, even the cows or grazing elements do not prefer them, difficult to tear off, but you can just roll like a leather. These are the cells or leaves are rich with a kind of a Sclerenchymatous cells which are called astrosclereids. They are star shaped sclereids. They are also living sclereid. Look at the arrowhead here, there is a living nucleus. So, these are nucleated living cells and they grow like stars and they make a network of stars, and this network of stars give them very high strength. These are the parenchyma ground tissues. So, on the parenchyma ground tissue layers, the stars just run across us and gives a strength, but this is not as strong as wood. The difference is that I told you sometimes back, and also I will tell you again later on today, that the many Sclerenchymatous cells grow together and make a clump of cells which you cannot bend them like what happens in the wood, but they are discrete, lot of parenchymatous in between. So, you can easily fold them, roll them. There are other cells, sclereids, they are divided based on their shape. Some of them are called bone shaped cells or osteoclerists, or there are rod shaped called macrosclereids, and these are the ones which are present in the other hard surfaces like in the seed coat. Many of the seed coats are very hard, and those are kind of sclereids provide the strength in those seed coats. Besides those sclereids, there are also fibers. Fibers play very important role in case of plant support as well as an economic part of the crops. This is a cross section of a plant. This is, you will see in the phloem there are many cells which are fibrous called phloem fiber, and these phloem fibers are present in many plants, like I told you in the cucurbitaceae plants, pumpkins, where you get phloem fibers. This is a cross section of a flax stem where you get the linen from, and these phloem fibers, there is a difference between now phloem fiber and the xylem fiber. Xylems or xylem fibers are highly lignified. In contrast, phloem fibers have a lignifications up to a certain range, not always, not yet fully lignified. Like among there you get linen, that is a type of a cloth, you get jute, that is a type of a phloem fiber. Jute is classically known as bast fiber or phloem fiber, linen is also similar thing, but they have a range from 5 to 15% of lignin. So, this lignin formation is not throughout. Like if you see a cross section of a soft tissue like this, what you will find, and stain for lignin, what you will find, these are the xylems, these are the cambiums, and then you have phloem fibers. The phloem fibers are also thick, but they are not lignified, whereas xylem fibers are thick and lignified. And not only that, what you will find, the xylem fibers are very compactly arranged one after another. So, this gives you a very strong wood structure. This is a structure of a jute plant cross section. What you will find, look at these structures, these are xylem. If you look at the xylem, what you will find that there are no parenchymatous tissues between the xylems, and all the xylems are arranged one after another as a ring of very hard tissues, and that gives you jute stick. After jute stick, there is a soft parenchymatous tissue, and then you have phloem and phloem fiber. Now, look at the difference between xylems and the phloems. Here are the phloem fibers, they are lignified, but they are not compact. There are cells which are soft. So, this is a phloem fiber, this is a phloem fiber, both are lignified, maybe up to 15% lignification is there, and then you have in between parenchymatous tissue. So, you can easily separate this fiber from this fiber if you can kill the parenchymatous cells and separate them out. So, this killing of the parenchymatous cells and removing the cell wall of the parenchymatous is a process called rating of jute. If you see a jute, this is how the jute sticks are, these are the jute fibers, and jute is grown like a big plant, it is grown, and then they are cut and floated in the water for 15-20 days, and during that time there is a lot of water and microbes grow in the water that take up the cellulosic components present in the parenchymatous tissue. So, the cellulose, pectin fibers which are present in the parenchymatous cells between the phloem, that goes off, and then the fibers become loose, and then one can separate out the sticks from the fibers, and that is how the jute is taken out and loaded for the selling in the market. So, this is what is a cross section. What, look here again, this is the compact arrangement of the jute xylem which gives you a jute stick. Many times the pith cells or parenchymatous cells, they die, they collapse, and you get a hole in the jute stick, and the jute fibers which are separated by the thin wall cells, and you can separate them out. These are the bast fibers from the linen. linen actually forms on the flax, where you get the jute fibers are made up of. So, now I have only few more minutes to discuss. So, today the last kind of fiber I will discuss as the cotton fiber. You see, the cotton is also fiber, jute is also fiber, but this is a difference. Both of them are actually are fibers, but cotton fiber is a cellulitic fiber, not a lignin fiber. So, cotton fiber is made, is an anatomically, this is a seed coat epidermal layer. So, if you look here, this is a growing ovule of a cotton plant, and this is the 0 day after pollination. That means, it is a very young. If you look under microscope, what you will find that there are large number of tiny hair like structure which is just ready to grow, and these all are trichomes. These trichomes are the ones which grow from the epithelial layer, from the epidermal layers, and this is the one which is within 3 days. So, within 2 to 3 days this fiber will come out and grow rapidly, and this is an one which is composed of, in contrast to the jute fiber, they are mostly cellulitic fiber, and their lignin component is very little over there, and they are mostly composed of cellulose. This is what a jute cotton fiber looks under microscope over here, as it is shown here, and this is a cotton big cotton mature cotton fiber which is grown over the years, and if you look not over the months, and this is what it looks under microscope. So, now coming to the overall summary whatever I told you today. So, this is just to another 5 minutes maybe I will summarize whatever I told you today for the students who might have joined late or listening now, that plant cells or plants are composed of mainly 3 types of cells: parenchyma, collenchyma and sclerenchyma. There are many different kind of deviations are there, many different types of development takes place over parenchyma. The parenchymatous cells are distinct by the presence of thin wall and roundish in structure, and they are the ones which contribute to the most of the growth in the young tissues. And when the parenchymatous cells die, they do not leave out much space, they shrink to a very thin component, and the parenchymatous cells provide support mainly with the help of turgor pressure. And most of the leaves what you see in the nature, the leaves which are erected against the gravity which holds high, and they are mostly due to the turgor pressure present in the parenchymatous cells. The parenchymatous cells sometimes get slightly elongated, like you have seen in the case of palisade parenchyma in the leaves, but quite often mostly they are roundish. Collenchymatous cells in contrast are mostly elongated cells. They are present in the hypodermal layer. They provide they are metabolically active and they provide support strength. So, you can see that collenchyma is a mixture of properties of both parenchyma and sclerenchyma. Like sclerenchyma, it can give support, and like parenchyma, it is very active in metabolism, and they are mostly elongated. They are present in the hypodermal area of the young stems and leaf petioles. And the last type is sclerenchyma. The sclerenchyma are the thick wall cells. They are metabolically less active. Their main job is to provide support, and you see what the furnitures you have around, all the wooden furniture, wooden floors, they are all made up of sclerenchyma, and the sclerenchymas are very economically important tissues like all the fibers come from Sclerenchyma, what you eat mostly are Parenchyma, and what are the ones which keep the leaves erected or the young stems hold on the large stems, they are all Collenchyma. So, I hope this lecture had been quite useful to you, and I hope it should, I mean many of you might have read already earlier, among them it must be a recapitulation for all of you, if not completely new to you, and I hope you have enjoyed it. Thank you.
With this note, thank you, sir. Thank you so very much for giving your precious inputs. Dear friends, if you have liked this lecture or if you have any queries regarding this particular lecture, then we always say that you can mail us for this. I am giving you our mail ID. Our mail ID is info.cc@nic.in. Here you can post your messages. I mean, you can mail us, you can send your feedback also, as well as your queries also. The next time when Dr. Nandi visits our studio, we would love to solve your queries here in the studio itself, and we would be meeting again with another session on types of plant cells. Till then, we take your leave. Take care. Goodbye. Thank you, sir. Thank you so very much.